करके मैं आरोग्य से
Hello, I am audible to everyone. Hello, dear team. Can you hear me?
Yes, yes.
Yes, yes. So, good morning, everyone. Today we are going to have our first session from 10 a.m. onwards, and we are waiting for the other participants to join, as the time has been given to them 10 a.m. And we will be having with us Dr. Ajay Kumar Chauhan from Sharda University. So we'll be waiting for others to join, and very soon, almost at 10:05, we are going to start the next session.
आवाज इसमें ओपन है क्या? इसलिए डबल डबल आ रहा है क्या? ये डबल क्यों आ रहा है? क्यों आ रहा है? साउंड कम करना पड़ेगा। साउंड कम कर दे, यहाँ से कम कर देते हैं, यहाँ से कर देते हैं। एक वो है स्लाइड्स चलो की मैं इमेज ही भेजता हूँ। मैंने उसमें हालांकि वो बना लिया हूँ। वो शेयर्ड वाली स्लाइड्स रहती है ना, जैसे अपन शीट और डॉक शेयर करते हैं, तो वैसे मैंने इसका प्रेजेंटेशन का भी एक शेयर्ड वाला वर्जन रहता है। हाँ, वो इसका ये वाला वो तो उसको क्या है, वो करना पड़ता है, एक्सपोर्ट करना पड़ता है, मैं भेजता हूँ। मैं तो इसका ना वही वाले को ना वो प्रिंट वो क्रॉप कर देता है। नहीं क्रॉप नहीं, उसको 2 मिनट लगता है, तो क्वालिटी वो वाली हो जाती है ना। अपन को कोई पाए नहीं, अभी तक नहीं आया रिस्पॉन्स। मैडम का कर दी 16 को। So, good morning, everyone. You all are welcome to the FDP workshop on the sustainable hydrogen production and the integration of hydrogen technologies. This is the second day of the workshop, and we are going to have our first session. In this first session, I want to welcome Dr. Ajay Kumar Chauhan from the Sharda University. Dr. Chauhan is currently working as an assistant professor in the Department of Biotechnology at SSBT Sharda University, Greater Noida. He holds a PhD in bioprocess engineering from IIT Roorkee with research focusing on the bioprocess engineering and strain development for biofuel production. He has over 3 years of industrial and almost 4 years of postdoctoral experience at ICGEB, where he worked extensively on metabolic engineering of E. coli and Penicillium funiculosum for enhanced higher chain alkane and bioproduct generation. His current research interests include biofuel production, algae-based CO2 capture, and application of synthetic biology and bioenergy systems. He has delivered expert lectures, published 20 plus research articles in peer-reviewed and SCI journals, and he is actively involved in the curriculum development and interdisciplinary research. So it's my pleasure to welcome Dr. Chauhan as our session speaker for this. So Dr. Chauhan, welcome and over to you. Uh, Wait for a minute. I think I'm unable to hear the voice.
Yes, yes, you are clearly audible to me.
Yes, yes. Now you are audible.
Okay, thank you so much. I am Dr. Ajay Chauhan. First of all, Dr. Ajay Chauhan, and presently I am working as an assistant professor at Sarda University, and here I am working as an assistant professor in the Department of Biotechnology, and here I am establishing the bioprocess engineering lab, where the fermentation process is of E. coli, fungus, and algae is going there. And we are also doing downstream processing here, downstream processing of certain products, whatever the industrially important products we are producing, we are doing the downstream processing. And we do confirmation using analytical techniques here. So first of all, I would like triple ITM Jabalpur and convener Dr. Tushar Chaudhary for conducting this faculty development program on sustainable hydrogen production and integration of hydrogen technologies. And this is a wonderful opportunity for me to interact with you all people about delivering my talk on biohydrogen production, pathways, manipulation, challenges, and future aspects whenever we are producing hydrogen. So, there for getting hydrogen in the linear and sustainable way, we always follow the biological route. So, in this biological route, what is important is that it's an alternate biofuel production, which is actually the environmental cleaner, and it is not going to affect your environment in any way. And whatever the researches we are doing, this researches is always correlated with the SDGs. So what is SDG? This is the Sustainable Development Goal, and this biohydrogen production comes under the SDG 7, where we can produce affordable and clean energy, because as current demand, you can see that the fossil fuels are exhausting, and we need to develop a process or technology which produces energy sufficient and clean energy. And this biohydrogen has a lot of analysis. So first, we start with the introduction, and as if we know that world is highly struggling with the severe issue of energy crisis for a long time now, and this is mostly due to the exhaustion of the fossil fuels, whatever present in the current scenario, and the reservoir for this fossil fuels are actually rapidly exhausting, because in India we are 16 billion people we are living here, so per capita consumption of this fuel is higher, and uh, that energy demand is fulfilled by using this fossil fuel. And as you know that as the gradual process of research is going on, so we need, as a researcher, we need to find a solution to produce a biofuel which is produced from the waste, organic waste, and we are not going to use the existing fossil fuel for the production of new kind of fuel. And as a revolutionary part of fuel, you can see that the biohydrogen production is one of the easiest and safest way. And once the technology get established, then world is going to fulfill with this hydrogen production. And as per the previous talk by the Dr. Tushar and other eminent speakers, they told you about how hydrogen is very important, because this hydrogen, as we are from the all the talks we know about, that it's a clean and it containing a very high energy fuel, which is around having the calorific value of 142 megajoule per kg. And this is the global need, because presently this hydrogen production mostly done by the fossil fuels, like natural gases are used for the production of this hydrogen. And this is almost the 40% of overall hydrogen production. And other like heavy oil, and from the naphtha it is around 30%. And there is another fossil fuel like coal, around 18% of hydrogen is produced. And there is only 1% of hydrogen is produced from the biomass. That biomass can be the lignocellulosic biomass or microbial biomass. So if we can use this biomass for the production of energy, it it means that we are using the renewable energy sources. And once this process is established, we can compare this process with the other existing renewable processes like electrolysis, steam reforming, and biological process. And once we know the cost analysis of all process, can we club this process together to get the higher hydrogen production? So we will discuss all this on this topic. So as you know that the biological process for producing the hydrogen, we can use a industrial waste, even a wastewater stream, like industry like tanneries, paper industry, textile waste. So these waste containing actually the COD, like COD is chemical oxygen demand, which already contains the high content of high organic content. So this high organic content is the source of carbon and hydrogen for the microorganism, and this microorganism going to consume this uh, hydrocarbons, organic hydrocarbon, and going to convert it into the hydrogen. And one of the major advantage of this biological process is these process are conducted at the ambient temperature, means 25-degree centigrade, and atmospheric pressure, that is 1 atm pressure. So we need to go for the very high temperature, like other chemical and physiochemical processes which are operated at very high temperature, like 200 to 300 degree, like pyrolysis and gasification. You need to go beyond 400 to 500 degrees centigrade. So most of these processes are operated at the normal ambient condition. And this process basically utilizes the microbial machinery for the conversion of hydrogen using proton exchange membrane fuels. And you can say that this hydrogen gas is produced from this. And this gases is free from the contaminant like carbon dioxide. Global hydrogen production market is valued is around 191 US dollar billion in 2024. And due to this government policies from the different countries, it is expected to grow by 2030, and it is also expected that the compound annual growth of around 10%, and the market is driven by the increasing demand for sustainable energy solution, and the potential of hydrogen is very large, and actually it is producing lesser carbon emission. So every country is adapting this technology and developing this technology, and as of now the global hydrogen demand in 2020 reached around 97 million tons with a 2.5% increase compared to 2022. And yesterday, in the introduction, some part of the Indian National Green Hydrogen Mission was discussed, and it discussed by the Dr. Aman. And this is the actually the policy why we are working in this area and to work in this area, because government is pushing to develop this technology, and this is the mission which is launched on 2023, and this is actually to become self-reliant Bharat by 2024 2047 in the in terms of energy, by achieving net zero emission of carbon dioxide and other pollutants, and this net zero target will be achieved by the 2070. And right now the target of 5 million tons per year of green hydrogen production by the 2030, and for this the government has allocated around 2000 crore of rupees, and this is all is done to expanding our existing technology and developing this technology. And the major key areas in this part is improving the manufacturing, means government is pushing the R&Ds and pushing industries to adopt this technology and develop this technology. So and for developing this we need to make hydrogen hubs, and for developing this technology the R&D requires the support from government as well as from the industries. So when we are
अभी
discussing
हम
about
टेम्परेचर बना के
biohydrogen,
रखे हैं,
so
तबीयत
this
खराब है,
biohydrogen is the
थोड़ा कम था।
best option among the all other biofuels like ethanol that government is blending with the petrol, diesel. So as we see that this hydrogen has the 3 times higher calorific value compared to conventional fuel like petrol and diesel, because it's around 142 kilojoule per this hydrogen is produced, and this is the petrol has the calorific value is around one third of this hydrogen. And another thing is that whenever you are using petrol and diesel, we know about that it is producing the NOx and SOx to pollute the environment. So hydrogen is the clean and environmental friendly, and this is this hydrogen can be produced from the organic waste, means whatever the waste coming from the industry, agricultural waste, we can utilize and exploit this waste for the production of hydrogen. The important thing that is this technology is offering the energy efficiency. So in terms of energy efficiency, this technology is very good, but the problem with this technology uh, is that the this technology require higher production cost compared to existing technology. And yesterday it was discussed that this hydrogen has very less density. So once the hydrogen is produced, it requires larger volume to storage for this hydrogen. So now come on the biological hydrogen production route. So as this talk about the biological hydrogen production route, so there are the majorly the 4 types of biological hydrogen production route. First is the biophotocatalysis. So bio, whenever you heard the term bio, it means from the living cell or living microorganism or living plant, and photolysis means it requires light energy to convert it into the into certain important product. So here is the androgen is an important product, and the blue green algae, cyanobacteria, and algae are the microorganisms which are used to convert this photon into your hydrogen. So this all the researchers are studied near the sea level, because the sea is containing a lot of cyanobacteria and algae, and once this sample of water is taken from that and analyzed, then it is found out that the lot of means some type of hydrogen gases are stored in that part of sea, and some other important byproducts are there. So the researchers are interested in this green algae and cyanobacteria, and that microorganism is isolated from that area, has the highest potential to produce this hydrogen. And next is the photofermentation. So photo means it requires the light, and fermentation means we are using the technology to converting, means using the microorganism and using its machinery to convert into the important compounds. So here is the hydrogen, and some other byproduct like acetate and format are also produced produced from this. So we can say that we can use this organic compound as well for the production of hydrogen. And third one is the dark fermentation. So dark fermentation is the technology we use the organic waste from the industry like municipal solid waste and other waste like energy crop waste, and we are actually performing the fermentation where we use a huge setup like for a fermenter, and there all the condition are controlled manner, so we can continuously produce the this biohydrogen. And next one is the hybrid type of system. It actually utilizes the electrochemical system where like microbial fuel cell technology are used, where you are taking the organic waste at one end and you are supplementing with the potent microorganism for producing this biohydrogen production. So from about two technologies, we know that the biophotolysis and photofermentation actually requires minimal amount of substrate and production of hydrogen, because it requires only the light
शाम के
and
2 बजे
carbon
निकलता हूँ मैं
dioxide from the atmosphere. If we can utilize this sunlight and carbon dioxide, we can produce hydrogen.
इमेज बुलेट
And
कैप्चर
now
होता
this
है
technology is actually uh, used for the carbon capturing from the various type of industry like the government has decided to restrict all the industry to emit their carbon dioxide into the environment, and this technology can be used for the carbon capture as well. The, uh, the advantage of this technology, it requires low energy inputs, and we are using the renewable substrate like carbon dioxide and
उसमें खोला
and sunlight for the
क्या
production of
बताइए
hydrogen.
मैंने
So,
ऑनलाइन दे दी
in the biological
मैंने
route, we need to know about what kind of metabolic pathways are used. So, uh, there are the metabolic pathways that can be divided into three. First is the biophotolysis, and that is categorized into two, where the
शेड
direct
वाला
photolysis is
प्रेजेंटेशन
happening.
एडिट
And
करते
in
वक्त मेरे
this
को
direct photolysis, what is happening? The microorganisms that are aerobic in nature, which is splitting the water and converting water into hydrogen gas. In the indirect photolysis means there are the system or machinery is present in the into the microorganism. In
कौन
so
बनाया
what is happening in presence of oxygen and the electron transport system, it is present
ये
in
मैं
the
बनाया
microbial system. Because first we need to understand a simpler pathway, like as a human being what we are doing, we are taking food and this food is converted into energy. So what kind of steps is happening? First is the glycolysis, where your glucose is converted into pyruvate, and this pyruvate further goes to TCA cycle and producing other type of ATP and reducing equivalent. And these ATPs are the energy, actually the energy which is utilized by the microbial machinery for the production of your important metabolite. And there is an electron transport system. There is a third thing, electron transport system. So in plant, what is happening? There are two type of photosystems are present: photosystem 1 and photosystem 2. And through the series of reaction, this photon light is passed, and this water molecule is split into the electrons, and these electrons are transferred, and further the enzyme, this H2ase is hydrogenase enzyme, is going to convert your substrate into hydrogen gas. Second one is the fermentation. In the fermentation, what we are taking the microbial algal biomass, and this microbial algal biomass is having the ability to convert your substrate into direct hydrogen. So there are two type of fermentation. One is the photofermentation, because if you are using cyanobacteria or blue green algae, it is actually utilizing the light. And through the catabolism process, it is actually producing the biohydrogen. Next is the dark fermentation. In the dark fermentation, its name suggests, because there is no light is present in the microbial machinery. So due to acidogenic and the fermentation process, the hydrogen gas will produce. And third one is the electrochemical. So there are some kind of changes are done in the electrochemical processes, and we can modify the process. One is the photoelectrical electrochemical process, where the photon is used for converting your organic waste into biohydrogen. Here, like specialized semiconductors were used for producing this biohydrogen. And next is the electrolytic conversion, means electrolytic conversion. Yesterday in the talk, there is the anode and cathode are there, and at one end you are supplying the organic waste, and this organic waste is utilized by the microorganism, and this organic waste is further converted into acetate and water, and this acetate is again going to use in by the microbial machinery for the production of biohydrogen. So this is the actually the simplest way to represent the metabolic pathway, how the biohydrogen can be produced. So in the first process, we know about what is biophotolysys. So the biophotolysys, it's an ideal method for producing the hydrogen, where the existing resources of the earth are utilized. So from this figure, you can clearly see hydrogen is supplied to the enzyme. This is hydrogenase, and this hydrogenase is converted into hydrogen. And when the hydrogen gas is produced, it is passed through the fuel cell, and you can produce the electricity or storage. It is depend upon the need. The advantage of this process is that there is whenever this process is carried out, all the CO2, whatever is supplementing, that is continuously removed from the system. So this is the process. And actually what is this process is doing, this process is actually splitting the water, means you are supplying water, and this water is splitted into hydrogen ion, and further the process is carried on. And the main challenge of this process, whenever the biological important product, so the important challenge, if their excess of oxygen is there, then you cannot effectively produce the hydrogen. So this type of process has low conversion rate. Now the type of hydrogenase we need to discuss about here, because depending upon the hydrogenase enzyme, because as a catalyst, we know about the turnover numbers, how the catalyst is converting substrate into product. So similar way, we need to know about the biocatalyst, because enzymes are known as a biocatalyst. So there are the three type of hydrogenases are present. One is the iron hydrogenases, and another one the nickel iron hydrogenase, and a third one is the metal free hydrogenase. So most how this process is happening, so there are the reducing compound like nitrogen and sulfur is there, and these compounds are actually performing the metabolism. So this iron hydrogenases are present in the green algae, and these are the efficient catalysts of hydrogenases, and performing the reversible oxidation of molecular hydrogen. And another type of nickel ferrous hydrogenases, these are present in the cyanobacteria. And other than this, there are traces of iron, sulfur, and magnesium ion also present. And these ions are all actually act as a cofactor and increasing the turnover rate of the enzyme. And the third one is that, as we discussed, the metal-free hydrogenases. So these are less existing hydrogenases. So uh, as we are using the hydrogenases, they are performing the catalytic reaction, as I discussed about the electron transport system and proton transfer system are present in the machinery. And when the system is present, there is some kind of channels are there. Whenever gas is passing through the system, so there are some gaseous channels are present. So these gaseous channel actually allowing this hydrogen means substrate to pass, and when once the hydrogen gas is produced, these channels are allowing this hydrogen to convert it into gaseous hydrogen. So in the another technique, there is the direct photolysis. So, as the name is known as direct photolysis, so this direct photolysis is the biological process which produces hydrogen directly from the water. And in this, the microalgae is used as a photosynthetic microorganism, and this microalgae has the capability to utilize the solar energy and converted this energy into the chemical energy. So in this reaction, what is happening? Two molecule of water in presence of light, and this is the common reaction we studied during our school time, is converted into hydrogen and oxygen. So here actually the water is splitting into the hydrogen and oxygen. The advantage of this technology, uh, this microalgae require can adapt the low light intensity as well, and still it is able to convert 22% of light energy. The hydrogen production in the green algae actually uh, takes place in the anaerobic condition, and the advantage of this condition, is further followed by the followed in the dark, and induces the activation of enzyme like hydrogenase and nitrogenase enzyme for the production of cell wall of the microalgae or any microorganism or plant, where there are the two type of photosystems are there, photosystem 1 and photosystem 2. This is the light energy which is falls on the this photosystem 1 and photosystem 2. The difference in these photosystems are they are actually absorbing the different wavelength of light for performing their reaction and converting their intermediate into products. So photosystem 2 actually converting water into oxygen and the hydrogen ion, and the series of reaction are performed, and it is passed to the photosystem 1, and then once the photosystem 1 uh, produces the electron, it is transferred to the ferredoxin, and there is the fate of the hydrogen where it goes. So due to FNR, it is goes to nitrogenase enzyme and producing the hydrogen gas like this. And if it goes to hydrogenase enzyme, it is going to produce the hydrogen gas. So the best way is that the hydrogenase enzyme is effectively producing the hydrogen, and once if this series of reaction enter into this route, it is requiring the NADPH and further energy, so the less hydrogen will be produced through this reation. So this is the
काय में भेजो
main, actually the
सर, वो
how
इंट्रोडक्शन
the
काय में
direct
भेजूं
photolysis is happening
ये
uh,
अभी जो आप मांग
using
रहे थे
microalgae.
काय
So
में
what
भेजूं
are the
व्हाट्सएप में
challenges
ईमेल
in this type of reaction,
ईमेल
as this enzyme, this reaction are requiring the need of enzymes. So these enzymes are actually sensitive to the oxygen, like hydrogenase enzyme is there, and if this enzyme is not is very sensitive to oxygen, means you will not get the hydrogen gas. So this challenge is overcome by the biological tools. If you can engineer the hydrogenase enzyme and make it uh, as a sensitive towards the oxygen, and you will get the higher hydrogen production. So for making this engineering of enzyme, there are different type of techniques are used, like site directed genesis and random mutagenesis. So what we are actually doing, because an enzyme is containing the various amino acids, so we are actually changing the base pair of this amino acid and checking that which base pair is the perfect fit for producing this effective hydrogenase or nitrogenase.
for this several mutagenesis process are already reported for doing the enzyme engineering and making enzyme more efficient, more catalytic effective, and more and converting and have the more turnover rate for converting the substrate. There are also the indirect photolysis. Actually in the indirect photolysis, what happens? This water is used by the microalgae, and this microalgae convert into hydrogen with the series of reaction. So first first target is that if microalgae is supplemented into in the system, so first we need to increase the biomass of that microalgae. And once the biomass concentration of algae is increases by utilizing the substrate, so further the dark anaerobic fermentation process is happening. So in the dark fermentation process, what is happening? So first we need to understand the reaction mechanism. So in this reaction, what is happening? 6 mole of water is reacting with the 6 mole of carbon dioxide into the supply. It is actually producing the carbohydrate and releasing the oxygen gas. So further what is happening? This carbohydrate again reacts with the water species and producing the hydrogen gas and acetate. And there are some amount of carbon dioxide is also produced. So 4 mole of carbon dioxide is actually utilized by the system and 2 moles of carbon dioxide released during this reaction. And what is further is happening in the indirect photolysis, this acetate is utilized by the microorganism in presence of water and again in the light because we are periodically supplying the light, and this is producing 8 mole of hydrogen gases and further produce oxygen output. So this indirect photolysis is applied in the microalgae and cyanobacterial system, but there are the spatial microorganism like Chlamydomonas and RT has been mainly used for the indirect photolysis and observed that this Chlamydomonas and RT utilizing the hydrogen enzyme system. Further, it is observed that this system is uh, actually increasing the hydrogen production rate and removing the oxygen, uh, which is actually inhibiting the hydrogen production. So, excess of oxygen during this entire photolysis fermentation process is also the hydrogen. So, we need to optimize the max is the biological hydrogen production by the fermentation. So, as all of you know that the fermentation process is a well-established process, and the key advantage of process is that we can continuously produce the hydrogen. In case of photolysis, what is happening when there is the light, we will get oxygen. When you are performing the fermentation and optimizing the biological processes, the hydrogen can be easily produced using the fermentation. And due to this fermentation, the hydrogen, whatever the hydrogen is produced, has more purity and efficiency. So in this fermentation, what we can use any waste stream from the industry, carbon sources like glucose, starch, and some other kind of carbon sources we can use. We can not use this glucose and starch. These are the compounds that we can use by all kind of people because there is always debate of food versus fuel. So we use always the waste stream like lignocellulosic biomass, also containing the cellulose and cellulose, and this cellulose and cellulose made up of this sugar. So we can perform the half fermentation, and there are some other wastes or industrial wastes are also produced, like if there is bad in the industry producing the food glycerol, which is glycerol is a highly reducing equivalent, which is easily utilized by the microorganism and and converting into industrial and food. So here is the hydrogen gas of the important product. So we utilize that machinery for the production of hydrogen. So theoretically, and what is the maximum theoretical yield using this biological hydrogen products and is that 53% of total oxygen demand can convert glucose to the hydrogen. So by the thumb rule, we can say that the 50% of hydrogen. So it is the thumb rule. But by the stoichiometry, we need to determine how much gas will be produced. So it's actually happening in fermentation. Suppose you are supplying the glucose as a carbon source. There can be other carbon sources which has organic waste, and there are the bacteria and microalgae are present. They are two catalyzing three type of reaction. These are the side reaction, which is producing carbon dioxide, main reaction, which is producing hydrogen, and other side reaction, which is producing methane, acetate, and other kind of hydrocarbons. So all this reaction is happening inside the fermenter because you are supplementing this fermenter with the substrate, microorganism. You are actually controlling the system using different level of pH sensor are there, oxygen sensor are there, carbon dioxide sensor are there, and there are the exhaust gases. This is the exhaust gases. These are the inlet gases. What are the inlet gases? Right. In the exhaust gases, you can couple with the your fuel cell where you can separate the hydrogen gases. So what is happening from the previous figure, you have seen that there are the hydrocarbon also produced. If you can utilize both hydrocarbon or product from acetic acid or nitric acid, these are actually then you can have some certain other carbon sources to the microorganism, and this microorganism utilizing this covered these acetic acid and nitric acid in the carbon. So this is the reason using in formation. And here fermentation we different form. In the food fermentation, like proper non bacteria like protococcus species are used. It is following the basic scheme where it requires a high amount of energy because we are talking about entity. So this all the reaction is carried out in the nitrogen machinery, which is utilizing the nitrogen gas and it is converting the ATP is converting into ADP and you will get the ammonia and hydrogen gas. So this is the reaction, these are the proton reaction during the reaction. So here what type of substrate you can use and what types of fatty acids, this you can skip now. So and these string you can use these substrate in the photofermentation and this photofermentation is completely on the light and anaerobic condition influencing the formula. Now, this nitrogen production is having the two stages now. In the first stage, they go up. I mean, absorption of light is there, and this due to this absorption, what is happening? The photodecomposition of the organic matter, and this organic matter is utilized by that microorganism. That microorganism is producing enzyme like nitrogenase, hydrogenase, and doing and these enzymes are really converting substrate to nitrogen. So despite having a lot of advantages, these type of system require high energy requirement and applying this nitrogenous rich we saw in the rating scheme, it is going it means it is utilizing the increased number of steps for catalyzing the reaction for the biohydrogen production. And in this other advantage of this is an advantage we have this the advantage of this monopoly this is producing other small molecules like acetate, lactate, and butyrate. You can use this molecule for the production of other industrial important products. So the one way we can produce hydrogen and we can take acetate, lactate, and butyrate and convert it into other metabolic like lactate can be used for lactic acid actually utilizing the carbohydrate substrate and converting it into into the hydrogen production. In this hydrogen production, the process which is not dependent on the light and yield high yield of hydrogen and the advantage of this process is for continuous production of both. It requires water. So the microorganism are Clostridium species and Enterobacter species are component species. And these microorganisms are actually following the acetate and butyrate pathway. And for controlling this adaptation, we need a simpler type of reactor and then doing substrate ranges can be used for the with this hydrogen. So what is happening? From the stoichiometric point of view, this glucose, sucrose, sucrose is also that molecule of glucose. Starch is also my already glucose and one for glucose made up from the glucose, right? And itself is also made from the glucose, right? So we can consider only single glucose unit, where one mole of glucose actually can produce means from this reaction. You can see this is that uh, molecular weight of glucose. The C6H12O6 is converted into the hydrogen gas. So from here you can say that the 12 mole of hydrogen can
सिंधु जैन आया
10 mole of the hydrogen. But in the top limitation that only 4 mole of the hydrogen
देवी का
because there is some thermodynamic instability is there. If because we know that if any reaction can be performed when that there is a negative form of this free energy there. The reaction is not having the negative or for a negative is having the negative gives it free energy or for performing the reaction. That reaction cannot be performed. This reaction having the limitation to 4 mole of hydrogen. So in the dark reaction, the key advantage is that you can use a type of micro if it is done by the six row and the hydrogen production can be faster. But other thing that is popular the opposite, oxygen sensitive opposite. If you supply the higher oxygen from the exterior source, you need to add the rinsing acid. Means if there is any kind of excess oxygen present in the system as well as inside the machinery, this oxygen is going to oxygen. Yeah. So what I will do then that is reducing at that time. As a scene, it's the answer, healing supply, the kind of oxygen supply for absorbing this excess of oxygen from our concentrate up.
काजल चौहान
And if it is still more by the competitive analysis envelope, then the hydrogen production is high. There are other species like Australia,
विक्की में था सॉरी किरण अरे वो अटेंडेंस अल्फा है कहीं था चले अटेंडेंस ले जाना फोटो खींच लो भाई खींच लेता हूँ किरण भाई किरण ये केपीसी की गुजर के पता नहीं क्या है कि देखो भी नहीं
So what is happening means what are the advantages of using it from means moving can be of temperature and the product cannot be means inhibited at a high temperature and maximum conversion of hydrogen is performed by this micro. From previous literature, it is shown that gas and heat and calculus is maximally producing 3.3 mole of hydrogen, and which is we can produce maximum theoretical yield of 4. If we convert 3 by 3 by 4, we will get 83% of maximum as around 33. And the maximum production rate or productivity of hydrogen gas, this microorganism is 8.4 millimolar per liter per hour and 4.5 millimolar liter per hour. So these are the capability this microorganism already. So how we can increase the dark fermentation rate? So in the dark fermentation, there are the certain process parameters like pH is very important parameter because most of the rate transfer from in between 5.5 to 8 and mostly 8.5 to and it requires the organic material. If organic material is not easily utilized by the microorganism, you cannot get the hydrogen, effective hydrogen gas. You need, whenever you are doing the dark fermentation, you need to supply continuous nutrition treated at the continuous fermentation for the utilizing where the microorganisms for maintaining cell growth as well as producing the hydrogen Another one is the temperature. If temperature rate is fluctuating, means this microorganism are very susceptible to the temperature, and their enzyme is also very susceptible to the temperature. So both can uh, be detrimental if temperature is higher or lower as problematic. So if you can maintain this condition, you will get the higher hydrogen yield. And the types of bioreactors are used are like batch, fed batch, and continuous mode. So the industry most preferably prefer fed batch and continuous mode for the high yield of this hydrogen formation. So this is all about the microorganism, but as a bioprocess or bioengineer, what we can do means as a bioprocess engineer, which can optimize the processes as a synthetic or microbiologist or synthetic engineer, genetic engineer, what you can do, you can engineer the microbe. Because if any microorganism is producing certain amount of hydrogen, so you need to uh, increase its potential for the production of hydrogen. So how you can do? In fact, how you can do? First, you need to design the pathway, you need what kind of enzyme, what genes are required for the production of hydrogen, and you need to analyze the catalytic rate of the switch, the catalytic rate of this enzyme. Once this design has been done, then you can do the building of that organism by using different techniques like CRISPR. This technique is utilized for the genome editing of that microorganism. Genome editing has been done. Even though you need to test that your this vaccine is effective or not. And further, this hydrogen production is performed by using various kind of you again design this BB here for the production. So in the metabolic engineering, what is actually happening? There's a principle of converted
अच्छा
in fibrinogen.
मैसेज जो मेरे
So this switch,
क्या नाम
this
है
result
क्रच
will
क्रच
be converted in lactate, ethanol, the sugar, milk, these enzymes in the technology and you move it from this now intermediate power. So what do you design a gene or design enzyme? So this is the hydrogenase enzyme, which actually introduced in the genetic in the DNA of the microorganism, and you are converting pyruvate maximally into format and this form of sugar. So natively, there are microprocessors which are using the same product, but once you have designed all the pathway, then you will get the maximal peak in the form of hydrogen. There is a possibility that if you are deleting certain kind of gene, then you will have recently the whole some kind of losing in the mother community. So you need to define again and again to get
लो
it.
अच्छा अच्छा बराबर
So similarly, you can do the genetic engineering in the microalgae as well. Here you can identify the gene like hydrogenase gene is there, which is going to be transport in the microalgae due to fermentation and presence of light. You are you are getting a synthesis and further you will get the byproduct from this microalgae. Once you have got the byproduct, this byproduct can be in the form of pigment, lipid, and protein. And this byproduct go to the residual biomass we do again for the fermentation and use for the harvest. In the two way you are performing the fermentation, first you are using the biomass and getting the other kind of product.
है
And once this biomass
ये
is in the fertility, you have
क्योंकि अटेंड का शुरू हो 190 जन पे रजिस्ट्रेशन ऑनलाइन कुछ लोग तो और
so
अटेंड के
how the hydrogen is
80 रुपए तो कोई
you can see that when you are doing the journey, there are the various type of traditionally designed options has been done, and there are the highest 2.11 mole of hydrogen is produced by the strain JWH1. This is the highest reported in the dark fermentation when the both are done at has done their genetic. So there is a in the dark fermentation there is a limitation. You can produce 4 mole of hydrogen gases, But for increasing the hydrogen production, means you can go up to the 12 mole of hydrogen production. So what can be done? So you can use the hybrid biological hydrogen production system. So this hybrid biological production system is utilizing the cell for converting this chemical energy into the hydrogen current in the form of can and easily observe that how much hydrogen gas are produced. So there are the technique like electrocoagulation is also used for the for the production of this hydrogen gas, where what you are using, you are treating polluted water, and one one end you are using the amount which actually dissolving this blend and producing the hydrogen gas, and this hydrogen gas are called in the form of bubble, and you are collecting this hydrogen gases in the bubble. So uh, from this figure, you can clearly see that this is here you are supplying the organic matter, and this is the anode where the organic matter is converted into carbon dioxide and hydrogen ions and electron. And once the hydrogen gases is liberated, it passes through the membrane, and once it is passed through the membrane, it is captured by the cathode, and you can check the potentiometer that you are getting the current in the form of right. You can check the hydrogen gas production in the form of that oxide. Fuel cell is there, and once the hydrogen gas is produced, you can pass three slides hydrogen now further from the sound. So my reaction is performing in this type of reaction, and I know what is happening. Like applying the current and acetate and water and stream of water are in the carbon ion and intermediate hydrogen and electrons are produced. And this is actually producing some, it is having some kind of electron potential like 0.187 volt of electron potential is there. And in cathode, you are actually taking the hydrogen gas out of that. So overall, this is given by the last equation, and you can calculate the electron potential. And when you are calculating the Gibbs free energy, if the Gibbs free energy is giving you the negative value, means it's the nature is and you can produce more hydrogen gas. So this type of process is what actually now modified from the as per the requirement and the function of hydrogen gas means if you are providing set side electricity to the system, so what is happening? There are the methane and oxygen which are producing in the system. They are not produced due to supplementation of the current. You will get the next problem. So now the some kind of feedstock, and we have already discussed what feedstocks we can produce, provide for the customer hydrogen, and we can do the pre-treatment methods for getting the maximum production of hydrogen. And you need to do the detoxification because when you are using the liposoluble grammars, you need to type of all that. So if you run this HMF, you need to remove for the liposoluble. So most of the biological process they are actually performed around the recording is integrated and into formal bacteria present within go up to 55 to 60 degree centigrade. To upgrade the operational pH is around 5 to 6.5. And the importance is hydraulic retention time means if you reduce the hydraulic and we need to optimize the substrate loading and material balance of multi-levels with our to getting the maximum production. And once the hydrogen gas is produced, we need to do the fast food separation because this gas is present in liquid, so you need to do the separation of the gases that are stored in there. And there are the several type of reactors also, like is here, fluid and sludge bank branded reactor are there. Forming these are type of reactor. There are other acrid and fluidized bed type of reactor. They are mostly used for the anaerobic type of fermentation. Other are the photobioreactors, which are used for the micromercury and bacteria. And as we are producing the hydrogen gases, most of all the challenges and bottlenecks of the process because the biological process has the low hydrogen gas release and productivity. So we need to understand the process, we need to understand pathway, we need to understand the catalytic efficiency of enzyme, how this enzyme are important for the this condition. Right. We also need to know if we provide a high amount of substrate or if there is high amount of byproducts are present in the reaction system. These are also are for inhibiting for the microbe and enzyme and which is always going to affect the health. So I will take most of the time I think I will take more than 5 minutes and I'll be to 20 that you for energy. But it's right now it is not competitive at all because from gasoline only $0.33 to much per kilogram. Yes, there is a possibility that you can direct calculator and the challenge of water, we can maximally convert this sunlight energy into sensitive in sunlight energy into hydrogen gas production. But this overall process is sensitive to oxygen and sunlight. If these type of features are not these type of features are not present in the reaction, then this is the problem for the entire production of hydrogen. But this process can be improved, like electrochemical processes, or if you find out low velocity which is already producing the highest hydrogen, or you can do the genetic engineering of that microorganism and couple that with technology with the other existing technology, and we can go up to the medical 95 this biological approaches. So from this technology, it is actually very costly. So if you're comparing with the fossil fuel or comparing from the other route and other chemical route, if the electric fast for these processes require R&D work, lot of R&D work. Once these processes are actually established, then you will get the clean environment with the good proof of it. So this is the way you can produce the low cost hydrogen production technology. You can follow that for the longer term and the term of
Today's
sustainability term.
session that
This
is from
you
3:30
can
p.m.
do
to 4:30 p.m.
what are the
Our
ideal applications
session
of
speaker
photo
is joining with us.
for checking energy? And this is a parameter in decentralized knowledge system, which is the parameter there. And don't forget that this is
देखते हैं बोले देखते हैं बोले खाने का टाइम हो
and right now we're actually pushing for the option of this hydrogen. Yes, sir. And producing a high hydrogen technology. And there are the recent advantages that massive in the like you can genetic engineering, synthetic biology, EIML, ML from the existing data, you can use that data, do the optimization. Once the optimization has been done, then you will get this technology compared with the. So now comes to the conclusion. Yes, hydrogen technology is a very promising technology. It faces a technical barrier, innovation, and right now the innovation is required. Technology be part of produce funding from the government as well as from the state. So thank you so much. Any questions? And you can contact me on that. And thank you so much, everyone, for listening.
So thank you so much, Dr. Chauhan. Now I'll ask the participants if you have any doubt. Okay. Amit Bhola has raised a hand. So
Good morning, sir.
Good morning, Amit.
Sir, can you show the slide on photolysis, biophotolysis?
Okay. share the slides.
Sir, here, what is the role of water? Because you told me the photo system is processed by the sun rays. So, what is the role of water?
Actually, the photo system is actually absorbing the light, means it is absorbing the particular wavelength of light, and all the metabolic reaction is performed. Means water is also act as a substrate for this reaction. This is absorbed by the system. In presence of that light, this water is split into hydrogen, ion, and electrons. So once this hydrogen ion is produced, this hydrogen ion is utilized by this enzyme, hydrogenase enzyme, which is already produced by the microorganism, and this microorganism is catalyzing the reaction producing the hydrogenase.
Okay, sir.
This is the way. Chlorophyll. Chlorophyll, which is containing different type of photosystem, photosystem A, photosystem B, and they are absorbing this light and they get activated absorbing this light and performing series of reaction to convert this water into hydrogen ion as well as electrons.
Okay. And sir, next question, thanks for the answer. In the process of direct photolysis, you have told that we can produce the hydrogen by the algae.
Yes.
So, the resources or and the island, it can be relocated at the Iceland or sea.
Actually, that photosynthesis, the concept of seeds that this process is already observed by the scientists. So you can right now you can see the algal system is used as a one pond system. Means this is
Yes, sir.
yes, one system is there. You can produce use this microalgae and go in the open system, and this microalgae has the tendency to absorb the light, absorb the carbon dioxide, and convert it into energy as well as hydrogen gas. But in the open system, you cannot easily store this hydrogen gas. This is the limitation. So that is why you require a device or bioreactor or fermenter for that.
So, sir, for that, this direct photolysis is only in the research work. It is not in practically not in application.
No, it's actually it's in one machine item. Means if you develop the like filter or system, any system, but you need a potent microorganism which can easily fix or which can easily grow in the low light system, because you will always not get the efficient light during some hydrogen gas. So this is existing, but you are making that microorganism efficient for producing the higher hydrogen production.
And minimize the
Minimize the
algae and the fungus.
yes, if there is less level of light is there, then you can get the same way hydrogen as the means natural microorganism.
So it can also be implemented at the Iceland or told on.
Yes, you can implement it because for developing this, you have to do the genetic engineering of the animal. Once the strain is developed, you can use that strain and test it at different level that how it is responding. And once you get the response, means why I told you PBTL means again you have to design the pathway. Means every system has different rule. If you go to means island, or if you go to central part of India, or if you go to sea, because microalgae present in the sea, they have different characteristics. So according to characteristic, you again need to design the pathway for getting the maximum.
Okay, sir. Thank you so much.
Okay, thank you so much, Dr. Chauhan, for joining us in this session and delivering your talk on biohydrogen production. A lot of work is going on this biohydrogen production, and a lot of biomass is available in India as well as in the world. And a lot of research is required to focus on this biomass to utilize for the hydrogen production. Thank you so much for joining us, and we would love to see you in our further coming sessions on the FDP workshop. Thank you so much.
Dr. Saran,
Thank you, sir. Now, I request all the participants, you can leave as of now, and you can join back in 10 minutes because we are going to start our session from 11:30. the next session will be delivered by myself only. So I request you, you can take a break of 15 minutes. Thank you. हाँ मैं ब्रेक, हाँ क्या वो ग्रुप में फिर जो मैसेज कर देते हैं कि ज्वाइन बैक कर लो। कई लोग क्या मोबाइल ज्वाइन कर रहे हैं, कंटिन्यू भी हो जाता है इस वजह से भी कर सकते। हेलो प्रशांत जी, झा जी, वो बैलेंस बता दिए रहते हैं। क्या बोल रहे हैं? तो बता दूं, 40,000 का मेरा लिमिट। हाँ हाँ,
यूसी मैंने बना दिया हुआ है उस फॉर्मेट में, एमपीसीएस की अलग फॉर्मेट में मैं आपको फॉरवर्ड कर देता हूँ, आप जो भी चेंजेस करेंगे आप उसी में करिएगा, और उसकी प्रीवियस वाली कॉपी भी मैं आपको भेज देता हूँ जो पहले आपको बनाया, ठीक है, दोनों भेज दूंगा तो ताकि आपके जो चेंजेस रहेंगे टिल डेट पे बनाना हो, ठीक है। हाँ जी, नहीं, ये अभी जैसे आज या कल तक के जो बना है ना सेटलमेंट तक टिल डेट मानेगा, पिछले बार तो ऐसे ही गया था, पुराना वाला तो मैं आपको भेज देता हूँ, और वो 40,000 का रिम्बर्समेंट वाला था वो सेटल हो गया। अभी तक तो कंप्लीट ही नहीं हुआ। भारतीय होम लैपटॉप पर है इसमें बाद में नहीं चीजें बढ़ती वो उसके कोई प्रक्रिया नहीं है, इसलिए विषय कैसे होता है ऑलराइट कैसे कर लेंगे हमारे वो बैठा करना होगा देखता है बहुत सारे हाँ ठीक है हाँ और लेके गए वो भी नहीं वहाँ बैठा है और फर्स्ट स्टेटमेंट एक ही लोग बता देंगे हिमांशु और ऑथर स्टेटमेंट वाले में जीओसी में क्या जमा किए थे सर
कौन सा
जनरल ऑफ एनर्जी स्टोरेज में हम लोग ऑथर
हाँ वो हम
देखो देखिए देख रहे हैं इसके ऊपर देखो डिक्लेरेशन में दोनों चीज का है ये दो अलग 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 है ना अच्छा ये यही जमा किए थे ना डिक्लेरेशन ठीक है ठीक है वही करना और ऑफर स्टेटमेंट ऑफर स्टेटमेंट
FIG लिखा है
है
FIG
ऑथर स्टेटमेंट ऑथर स्टेटमेंट जब फाइल अपलोड हुआ
अच्छा अपलोड करते तब की
हाँ उस टाइम क्या जमा किए थे वो बताओ तो ऑथर स्टेटमेंट
एक्नॉलेजमेंट वाले कंप्यूटर में
ये पंखा काहे बंद
वो सर ऑटोमेटिक कुछ बंद हुआ है कुछ बाहर मेंटेनेंस चल रहा है
ये पंखा चालू कर रहे हैं
अरे यहाँ से नहीं हुआ इसको कुछ
रिवीजन में हो गया देखो डिक्लेरेशन और ऑथर स्टेटमेंट क्रेडिट ऑथर स्टेटमेंट conflict of interest. There is no conflict of interest.
देखो ऑटो है और शायद लाइट आइडिया कुछ का प्रॉब्लम है।
ये तो हो गया ऑथर स्टेटमेंट में वो रहता है दुबे ये क्या बोलते हैं उसको नहीं कवर लेटर नहीं रहता वो कि कौन कौन क्या क्या कंट्रीब्यूशन किस डायरेक्शन में
क्रेडिट
किया
ऑफ स्टेटमेंट
हाँ है हाँ जो तुमने जो वो शेयर किया है पीडीएफ वाला उसमें है कि नहीं देखो तो एक बार उसमें है कि नहीं देखो तो एक बार हम्म
ये अपन पहले ही डाल देते हैं रिवीजन पे
हिमांशु को मेल हम्म
आपको भेज दो हाँ
एक मिनट ऊपर ऊपर मुझे सुनते हैं
So again, good morning to all of you, and we can wait for 2 more minutes so that other participants can join.
ये नंबरिंग अपने आप आएगा कि हाँ हम लोग तो डालना है इसमें? Declaration of Freedom of Information. Nonsense speaking डालना है इसमें? लिस्ट ऑफ टेबल भी भेजो, टेबल में भी कुछ चेंजेस किए हो क्या?
So, very good morning to all of you. My name is Aman Singh Rajput, and today on this FDP workshop presentation on the sustainable hydrogen production and hydrogen integration technologies, I am presenting my uh, presentation on the energy, exergy, and sustainability analysis of a diesel engine with biodiesel, nano additives, and the hydrogen gas. This is a brief introduction of myself. So, I'm working in the field of thermal and automobile engineering, specifically in diesel engines, biodiesel, nanoparticles, energy, exergy, sustainability analysis, and at present I am working in hydrogen production and its utilization in the diesel engine. So this is the background of 2%. So, but a lot of options for the renewable energies are there. Like we have the solar energy, which is prominent in the area of renewable sources, and the nuclear energy has been explored, but at the same time, nuclear energy is very hazardous. So, renewable energies are very clean and clear, and they can produce no emission when they means produced or when they converted from one form to another form. So that is why a focus is there on exploring the renewable sources of energy for the utilization in the appliances and different types of applications. Moreover, the internal combustion engines are vital in transportation and industries, and because their efficiency and power is more. At the same time, uh, when the environmental concerns are there, then we can see that a lot of emissions can be seen in case of internal combustion engines, specifically in petrol and diesel engines. So the diesel and petrol has been used for a long time in the internal combustion engines. So biofuels that can be made from the renewable resources, that can be an alternative solution for the uh, internal combustion engines, both for the diesel as well as petrol engines. So, in lieu of this, India has proposed its national policy on biofuels on 2018, and its national green hydrogen production, as I told earlier yesterday in the morning session. In 2023, India's national green hydrogen mission is proposed. They are proposed to contribute or towards the um, energy independency and to achieve a net zero goal emission. So the alternative biofuels can be ethanol, biodiesel, and biogases. So ethanol can be replaced, means ethanol can replace petrol, gasoline, or LPG. Biodiesel can replace diesel. Biogas natural uh, can replace natural gases. So the focus of this particular study is on the biodiesels. So diesel and biodiesel, what is the main difference between the diesel and biodiesel? Is you can see that diesel is basically a hydrocarbon chain having only the hydrogen and carbons, whereas the biodiesel contains hydrocarbons as well as an additional oxygen is there in the biodiesel. So this oxygen plays a role in means differentiating the properties of the biodiesel from the diesel fuel. And also when it is used in the diesel engine, then due to this difference in the chemical structure, the performance of the diesel engines is varies. So biodiesel is proposed for diesel engines, and a lot of study is going on diesel engines because heavy vehicles, if you have seen that in heavy vehicles the diesel engines are there, and diesel engines can only be used in the heavy vehicles due to the higher compression ratio and higher energy density of the diesel engines. So we cannot use the petrol engines for heavy load applications. So the diesel engines which are used for heavy load applications, they are using diesel engines. They are having very less efficiency and very high specific fuel consumption. यदि आपने देखा हो तो डीजल इंजन जो हैवी व्हीकल्स में होते हैं उनकी आपने माइलेज देखी हो तो माइलेज उनकी यूजुअली 2 3 या मैक्सिमम 5 लीटर पर किलोमीटर होती है, सॉरी 5 किलोमीटर पर लीटर होती है. तो so this is the basically means edible oils or the waste biomass. Whatever the content is there means the free fatty acids, um, जिसमें भी आपको free fatty acids मिलेंगे जिन भी biomasses में. So you can use them free fatty acids or the lipids in the triglyc TGS triglycerides for the conversion into the biodiesels. So we have different types of generations of the uh, biodiesel. We have named them as per their means properties. Like if you are producing the biodiesel from the edible biomass, then it is called the first generation of the biodiesel. And when like sugar beet, sugar cane, wheat, corn, these are the edible biomass. And with the oil of these biomasses, if you are producing the biofuels or the biodiesel, you can name it the first generation biodiesel. Then the second generation biodiesel is made from the non-edible biomasses. So non-edible biomasses, which are usually not consumed by the human beings. So they are named as second generation biodiesel, and nowadays, before a few years back, Jatropha is proposed for a potential biomass for conversion into the biodiesel. So that Jatropha is specifically means in Hindi it is called Ratan Jyot. तो रतन ज्योत और जो जेट्रोफा जो होता है आपका, that is the second generation biodiesel. It is made from the non-edible biomass, but it is somehow non-edible, and it can uh, uh, basically the first and second generation biomasses require the biomass to grow, and we require land and water. So basically we have to cultivate them. So there is with um, first generation of biomasses, there is a challenge of food versus fuel. And in second generation of the biodiesel, you have the competency between the fuel versus land. So here comes the third generation of the biomass. The third generation of biomass, which is the waste biomass. You can call it like it is undesirable desirable products that is grown in the earth, and but they have a lot of potential or the nutrients or the different types of enzymes, lipids that can be converted into the biodiesel. So that comes under the third generation of the biomasses. So microalgae and macroalgae are basically the third generation of the biodiesel. So because we don't algae जो काई होती है आपकी या शैवाल होते हैं इसको आप you don't need it. They are unwanted products. So when they grown, but they have a high amount of lipid contents and that can be used for the conversion into the biodiesels. And last comes the fourth generation of the biodiesel, and which is being researched, and it is proposed that it will be the genetic modification of the present algae biomasses. So basically, this fourth generation of biodiesel is not yet tested, but it is researched, and most probably in upcoming few years, you will be see the fourth generation of the biodiesel. So this is all about the biodiesels. Then it is basically non-toxic in nature and produce less emissions as compared to the fossil fuels. But when it is researched with the diesel engines, then we have got some less efficiency as compared to the conventional diesel engine or with the use of conventional diesel fuel. So what the researchers are doing, they are proposing a new technologies like using nanotechnology. So they are using different types of nanoparticles blended with the biofuels in a specific ratio. And by doing this, the efficiency and other parameters can be analyzed like a specific fuel consumption emissions. So using these nano additives, we can improve the performance of the diesel engine. At the same time, when the gaseous fuels are blended with the uh, means blended, not blended, when the gaseous fuels are supplied to the engine in a different configuration, they can be used as a supplemented fuel and increase the efficiency of the diesel engines. The next is the energy exergy and sustainability analysis. Why we need the energy exergy and sustainability analysis? Because whatever the uh, efficiency we are getting, that is basically the outcome of the engine, and it is based on the first law of thermodynamics. Means this energy is converted into this energy. But at the same time, what quality of energy is converting into the useful work and how much potential is still there in the system, that can be analyzed by the exergy analysis. So exergy basically deals with the potential which a system have it is if it is brought into the equilibrium condition with the atmosphere or in the environment. सो आपने 1 लीटर पेट्रोल या 1 लीटर पेट्रोल डीजल जलाया, उससे जो भी आपको इफिशियंसी मिली वो आपने वो आपकी एनर्जी एनालिसिस के अंडर आएगी। लेकिन जब आप उसको थियोरेटिकली कैलकुलेट करेंगे या अ, जैसा कि कार्नोट साइकिल आपको बताता है, कार्नोट इंजन बताता है कि दो टेम्परेचर डिफरेंस के बीच में जो इंजन आपका वर्क करता है उसकी इफिशियंसी जो होती है मैक्सिमम होती है। सो व्हेन यू कैलकुलेट दैट इफिशियंसी बिटवीन द टू टेम्परेचर्स ऑफ source and sink, then you will get the maximum efficiency that can be derived from a system and how much it is deriving. So that comes under the exergy analysis, that how much potential is still there in the system, or the potential that can be extracted from the system to effectively utilize the input energy. So that comes under the exergy analysis. And then the sustainability analysis means how much of the energy you are giving to the system and how much energy you are getting from the system. So the ratio of that is called the sustainability analysis, means the conversion efficiency. Basically, you can say the conversion capacity or the maximum utilization of the available energy with respect to the uh, output energy. You can call it as sustainability parameter. So this is the research motivation, that we are having the environmental impact, the we want to improve the performance, and we want for sustainability for a long term viability of the fuels. So the objectives of this case study which I am presenting here is to perform a comprehensive analysis on a diesel engine with biodiesel and nanoparticles to analyze the combustion performance and emission analysis. Then to characterize the biofuels and see the results obtained from the UV, FTIR and gas chromatography. Then to perform the experimental analysis on the energy, exergy and sustainability aspects of the diesel engine with biodiesel and nanoparticles under the varying operating conditions. So first we go with the biodiesel production. So biodiesel production basically deals with the first choosing the feedstock. So here we have chosen the third generation of the biomass, that is the microalgae, and specifically it's Chlorella vulgaris biomass. So this is collected in our lab. We have made a small amount of means biodiesel. So the process is involved in it is harvesting, means collecting and drying of the algae, then the oil extraction. So in this specifically, this specific case study, we have used the sock slate extraction for the extraction of the oil from the uh, microalgae biomass. Then we went for the acid catalyzed esterification and then base catalyzed transesterification. So basically, transesterification method is used to generate the biodiesel, that is called fatty acid methyl ester. But when we directly go for the transesterification, that is base catalyzed transesterification, there may be some problem of the soap formation. That is why to avoid the soap formation, we first go to the acid catalyzed esterification of the free fatty acids with methanol under the catalyst, that is sulfuric acid. So we got the fatty acid methyl esters plus water. Then we go for the transesterification. So whatever the triglyceride we have we got from the um, esterification, then it is further went for the transesterification. So the transesterification is again done with the methanol, but here we have used the base catalyst, that is the potassium hydroxide. So we got the fatty acid methyl esters and glycerol as the byproducts. So the glycerol is heavier in nature. It is having the higher density, so it settle down at the base of the um, means the mixture. And the upper part of the mixture is the fatty acid methyl ester, which is our biodiesel. The reaction time which we have achieved, we have tested with you that collecting the biomass, then wet algae is there, then we have dried it, then dry micro algae powder is made. Then we went for the oil extraction, then the oil transesterification, then finally we got the biodiesel. So these are the samples we have prepared. So diesel is there, pure microalgae biodiesel is there. Then we mixed in a quantity of 20 percent of the biodiesel with 80% of the diesel fuel. So we have tested it because in several studies it has already been proposed that using up to 20% of the biodiesel, so no engine modifications is required. So we can use up to 20% of the biodiesel, but beyond it, you will seeing some um, disturbance in the performance of the diesel engine. So we can use up to blending of 20% of the biodiesel with the diesel fuel. Then after blending, after taking the B20 blend, B20 means that 20% biodiesel and 80% of diesel fuel, we have used titanium oxide nanoparticles for its blending with the uh, B20 biodiesel blend. So we blend it in a quantity of 100 parts per million, means for uh, 1 liter. For 1 liter, we have used 100 ppm, 0.1 gram of the nanoparticles were used, and they were blended with using the ultrasonicator bath. Uh, and when the blended fuel biodiesel is achieved, then we go for the testing in the diesel engine, that how it performs with the diesel engine parameters. This is the experimental setup we are having in our lab. That this diesel engine is basically a Kirloskar made uh, diesel engine which is having 661 cc of engine, 661 cc of engine, and 3.5 kilowatt of rated power. Then it is water cooled engine, single cylinder engine is there. And it is connected with the flue gas analyzer. We have ABL 444 gas analyzer there, and it is connected with the computer unit to analyze the data which is performed by this diesel engine. So these are the engine specifications. I already told you it is made from Kirloskar. Then 3.5 kilowatt is the rating, and other parameters which is it is having, it's shown here. The boundary conditions for the uh, testing was displaying here. So basically we went for the simulation as also, and went for the experimentation also, and then we validated the results of the simulation as well as the experimentation. So here I am presenting only the experimental results. So compression ratio which we have used is 17.5. The engine speed was maintained at 1500 rpm. All the other conditions like pressure, temperature is same as the atmospheric conditions. The injected timing of the engine and injection pressure is 220 bar. Then the inlet and exhaust valve timing is displayed here. This is the yes, it is the rated timing of the engine which we have used for our testing. These are the measurement uncertainties which we have taken under consideration, because whenever you go for the experimentation, so यदि आप सिमुलेशन करते हैं तो आपको एक सा रिजल्ट मिल सकता है हर बार जब भी आप सिमुलेशन रन करेंगे विद द स्पेसिफिक बाउंड्री कंडीशंस। लेकिन जब हम एक्सपेरिमेंटेशन करते हैं तो हो सकता है, क्योंकि it has a lot of effects from the environment and other parameters, so there may be some uncertainty in the measurement. So based on the specifications of the measuring parameters or the uh, devices, we have taken here the uncertainties of different instruments. So, and based on that uncertainty of that instruments, we have calculated the total percentage uncertainty. And when it is calculated, it is about 2%, 1.78%. So, whatever the data it shows, whatever the data we are getting, they are having 2% of uncertainties in the measurement. जो भी हमको data यहाँ पे मिल रहा है, वो या तो 2% कम या 2% ज्यादा हो सकता है, based on the considering the uncertainties in the measurement. This is the energy and exergy analysis calculation. So how the energy and exergy analysis is calculated? So basic science behind it is the in both energy as well as exergy analysis, we considered the exergy going into the system and the uh, utilization of that energy in different parts of the system. So when we consider energy analysis, we usually go for the whatever the energy is going inside the system and how much energy is distributed with different types of system, like how much energy is converted into useful work, that is shaft work, then how much energy is taken away by the cooling water, then how much energy is taken away by the exhaust gases. So this is basically comes under the energy analysis. In exergy analysis, we considered how much available energy is is still there in the systems which I have mentioned here, like whatever the available energy is there in the shaft power, in the cooling water, in the exhaust gases, what available energy is there which can still be utilized for further conversion into some useful work. And based on the this exergy and efficiency, we have calculated the sustainability index. So these are the some of the properties of the fuel samples which we have used in the study. So I should elaborate the terminology or the fuel sample designation. So CB is used here for the 100% of diesel, means biodiesel is zero in there. Then CB20 means biodiesel is 20% and 80% is uh, chlorella vulgaris biodiesel. Then CB20 Ti means we have blended titanium oxide nanoparticles in a quantity of 100 ppm. CB20 HHO means we have supplied 0.3 lpm of hydrogen gas to the diesel engine uh, with uh, means the blended biofuel, that is the CB20. And again, when the last one is CB20 TiHHO means we have supplied the hydrogen with the blended fuel of biodiesel, diesel, and nanoparticles. So then this is the characterization of the biodiesel. Biodiesel is characterized. So we have tested the pure biodiesel and the 20% blended biodiesel and the uh, diesel fuel. So here we are getting this fluctuations in the diesel fuel, that is due to the impurities present in the diesel, because diesel was purchased from the market and there may be some contaminations in it. That is why we are getting here the fluctuations more in the case of this black region, and this black region shows the CB20 blend means CB20 CB0 means diesel fuel. So by using this UV spectroscopy, so basically this 200 to 400 of the wavelength presents the ultraviolet range, and 400 we are getting the higher absorbance in the biodiesel, which suggests the presence of the fatty acid methyl esters, which absorbs the which is absorbed in the strong UV region. So due to the double bonds present in the biodiesels. So low fluctuations in the biodiesel indicates a more consistent and homogeneous composition of the biodiesel, and very less contamination is there in the biodiesel blends, as I already told you. The lower absorbance and high fluctuation in the diesel suggest the dominance of the saturated hydrocarbons and the aromatic compounds like benzene in it. Then the reduced absorbance peaks and noise suggest more efficient conversion of the cleaner feedstock in the microalgae biodiesel. So basically, this is all about the UV spectroscopy, which tells about the photochemical absorption or the presence of the fatty acid methyl esters in the fuel sample. Then it is a there is a characterization of the biodiesel in which we have carried out the FTIR spectroscopy. So FTIR spectroscopy basically deals with the presence of different types of functional groups that may be present in the biodiesel. So by analyzing the graph with the standard graph, you can conclude that how what type of bonds are present in the fuel sample, and then you can predict the properties of the fuel samples, that how it will perform. So that is why we first went for the FTI spectroscopy. So we are getting here ester bonds is are present here. The triglyceride are converted into the methyl esters. So there is a presence of CO means double CO with double bond stretching. Then CH stretching confirms the presence of the hydrocarbon chains, and the oxygen and hydrogen bond presents the some of the or excess methanol or some water contents can be there. So the OH stretching presents usually it, but we are getting a very less amount of it. It means it shows the purity of the biodiesel. Means basically हमारा target biodiesel तो बनाना है, but this GCFID it identifies that what type of fatty acids are present in the sample and what type of fatty acids are converted into the fatty acid methyl esters. So these are the some of the results which we got from the result of the diesel engine testing. So when we tested them, so here I've shown in the left side the energy distribution, and on right side I have shown the exergy distribution. So the energy distribution tells how much energy is distributed inside the engine system. Exergy distribution shows how much energy is still uh, there, or the available energy of the engine system. So likewise, whenever you see a trend of any specific fuel sample, so we have tested it with 4 different loads. So whenever you go for the specific fuel, so you will see that the efficiency utilization is increasing the engine load. So when you increases the engine will perform at part load conditions. So here the lower characteristics show the efficiency conversion of the fuel samples. So, like if I talk about the diesel fuel, then only 19.76% is the efficiency of the diesel engine. Means if 100 kilowatt of engine is giving to the engine, then only the 19.76 kilowatt we are getting as a output, and rest 80% of the energy is either associated with the cooling water, or either associated with the exhaust gases, or it is destroyed as in the inside the system in friction, in radiations, etc. So when we see this again at 100% load, then we can see that 35% of the input energy is converted into the output energy, means CW, EG for exhaust gases. So